So when you set up a manufacturing process, you have to run it through with cells to be sure that it works and does what it's supposed to do. And at the time we started this manufacturing process, the only cells we had available were what we call the so-called research cell bank, which was made for the PharmaCyte product. So we set up the whole manufacturing process based upon the research cell bank. That means we tested how fast the cells grew in particular conditions. We tested the right environment for the cells in terms of the temperatures, the speed of movement of the cells, etc., to give the best possible product. Parallel to that, PharmaCyte was moving the research cell bank to what we call the master cell bank, which is an upscaling of the research cell bank, so we have many vials which are all identical of a cell bank which we can use for manufacturing. Now during that process, and this is quite normal in biology, the cells can change slightly. And small changes in parameters in the cells make huge differences to the way the cells grow. So what we found when we got the master cell bank back is that many of the parameters we'd set for the research cell bank were not spot on. And so although we could start making a product, the cells didn't grow quite as much as we needed to fill out the capsules, for instance. So then we had to go back and we had to tweak the whole process based upon the master cell bank. And during that process we were able to improve the speed of cell growth, the way the cells grow inside the capsules, specifically for these master cell bank cells. And now we have a product and a method which is able to fill the capsules and able to make a reproducible product. And that whole process it's lengthy, it's not easy, but I think it's been worth it because now we have an excellent product at the end of the time. It's a robust product and it's really properly doing everything we want it to do. After the manufacturing is finished and we've filled the syringes and we've frozen them down, the next step is to test them. We call that release assay testing to ensure two things. One, that the product does what it's supposed to do. In other words, that it has the activity we need, the ability to convert ifosfamide into its active component. And that testing is done here, in-house at Austrianova, Thailand. The other thing that has to be tested is that the product is sterile, free from what we call adventitious agents, that means anything we don't want in there, and at the same time free from particles. All that testing is done out of house. We send this out back to Europe to accredited labs that check, using US FDA norms, that the product is within spec for safety. When we have that information, we will then write a so-called certificate of analysis, a COA, and that COA will accompany the final product to PharmaCyte. And PharmaCyte will then receive the product, check it in, and move it towards the clinical trial. We are involved in the IND production in terms of the actual manufacturing process has to be documented, and so we're involved in helping write that section because we're doing the manufacturing. Obviously, with our experience from before, we are also involved in writing the other sections which have to do with the past clinical data and the assays that are used and why this particular product is particularly well suited to treat this particular kind of cancer. Right. So the data has to be included in the IND that's submitted to the FDA, and once that data is available, it will be slotted in, and then the data package will be sent to the FDA. There's 20,000 cells on average in each capsule, and we have 300 capsules for one dose. So you're talking about 6 million cells all having to do the same thing at the same time and stay alive and do whatever they're programmed to do. That is quite a challenge. And if you look in the world at how many biologicals have been approved, there's not many. And the reason for that is because it's very hard to make these. The fact that we have a product which is in its own protective little home. Most cellular therapeutics today are cells which are just given loose. They're just injected as cells. And cells will then wander off and do whatever they want to do. They don't stay together and work together in consort as one small sort of island. In our product, we have the cylinder box, which keeps the cells together. The cells are all doing the same thing which they're programmed to do at the same site where we've injected them, at the correct site. Further, as I mentioned previously, we have a freezing possibility here, which we don't normally have with cellular products. So we can actually freeze our cell-in-a-box product, and we can store it for long periods and ship it around the world. So these are quite important aspects. Some of the cellular products we have today are more like treatments. You go into hospital, you get some sort of treatment, and then you come out of hospital again. But basically the cells are made in hospital for you. There are very few products which are actually made in advance, kept, used as the patient walks in through the door. And cell-in-a-box is one of those. This is the future of medicine. This is the new generation of how we're going to be able to treat diseases, not only cancer, but a whole plethora of other diseases. We're going to be able to take cells, we're going to program them to do what we want, and we're going to put them back into patients' bodies, and they're going to react to signals in the patient to produce medicines in the right way. So what PharmaCyte is doing, and what we are doing with PharmaCyte, is we're actually taking the first few bold steps for the new medicines of the next generation.